Thank you very much. I'm going to present a critical reevaluation of a Nature paper which claimed that translation is the key step in determining the differences between gene expression, while we found that they have underestimated the transcriptional importance. So people agree that the steady state of protein concentration in the cell are determined by the effects of 4 steps: the rate of transcription, mRNA degradation, translation, and protein degradation. Therefore, the differences in the expression levels between highly expressed genes, low expressed genes, and non-expressed genes are the combined effect of those 4 steps. So Schwanhaus et al. Published a paper in Nature in 2011, 2 years ago. They provided the most detailed analysis of decomposing the combined effect into 4 steps. They measured the half-lives of protein degradation and mRNA degradation, and also they have protein data from label-free mass spectrometry and from RNA expression data from RNA-seq. So in their paper, they used the right figure here to show that the scatter plot between RNA levels and protein levels have a very poor correlation. Sorry, I don't know why the text labels here is not showing correctly. And so therefore, they said that RNA level cannot explain well the protein levels. And there are other works who also use this kind of scatter plot correlation analysis to say that translation is a more dominant step in determining protein levels. However, we found that all these studies didn't consider the errors in the measurements. So we compared the protein estimates in Schwanhaus et al. For about 66 housekeeping proteins, including ribosome, nucleosome, and some transcription factors. And showed that their large-scale label-free mass spectrometry measurements compared to previously reported individual measurements, the ratio is not 1. We can see that the ratio is very close to 1 for the highly abundant proteins. However, it's much greater than 1. So the ratio is between individual measurements versus large scale. The ratio is much greater than 1 for lowly abundant proteins, suggesting that the systematic large-scale protein estimates are underestimates for lowly abundant proteins. And also, we look at the data in Schwanhaus et al. And found that their spiking proteins as standards only cover the high concentration area rather than the low concentration area. On the other hand, in their protein estimates, about 90% of the proteins are lying in the lowly abundant area. So this is a problem. Therefore, we corrected their large-scale protein abundance estimates by using the 66 individual measurements from earlier studies. So after the correction, we see that the ratio in the lower left panel is more close to 1. And with the corrected protein estimates, and we re-plotted them against the RNA data, we see that the scatter plots on the right, if you compare the upper panel and the lower panel, you can see the scatter is more concentrated. And also the line of best fit is much better. The correlation is higher. So we think that this is one issue the paper didn't consider. We also consider another 2 sources of error that is lacked in their paper. One is the stochastic protein measurement error. They have 2 replicates of mass spectrometry data. However, in their study, they used the mean data from the 2 replicates without considering the stochastic error. Another source of error is from RNA-seq. So we all know that RNA-seq suffers from, say, biases like GC content bias. And this is the right figure is a figure from their paper in which they compared RNA-seq data with nanostring, which is a more accurate RNA quantification method. Because when they compare nanostring with spiking RNA standards, the correlation is very close to 1. However, this is not true for RNA-seq. So therefore, this plot shows that there are systematic measurement error in RNA-seq that they should also consider. So given those data, so we are comparing, so we use a statistical analysis called analysis of variance, short as ANOVA, to decompose the measurement error, which should be here. So this is the variance of protein error. Okay, so I'll just describe it. So we decompose the variance in measured protein into different components. So we remove the variance in the stochastic protein measurement error from the protein variance. And we remove the variance from the RNA-seq error from the measured RNA variance. And therefore, we can derive the variance of true protein levels and variance of true RNA levels. And from that, we can re-quantify the percentage contributed by transcription. So this is our result. We also use the data, the half-life data of protein RNAs from the Schwanhauser paper. So based on our reanalysis, we can see that the percentage contributed by translation, which is the red part, has been shrunk. And the percentage contributed by the RNA, by the transcription part, which is the green part, has been enlarged, suggesting that it is not, so the Schwanhauser paper doesn't have enough evidence to claim that translation is the dominating step here. Another thing we didn't consider is, another thing didn't consider in Schwanhauser paper is that they only considered about 4,000 genes whose protein and RNA expressions are high. However, based on another paper on Hebenstreit et al., so in their paper, they found that about 40% of the genes in a mammalian cell are not expressed. So the number is about 8,000 genes. So given those genes with no RNA and therefore no protein, they should be they show that transcription is a very important factor in determining protein levels. However, those genes are not considered in the Schwanhauser et al. Paper, because for this part, the percentage should be 100%, right? And so, therefore, we think this is one thing that should be considered. Another thing should be considered is the systematic measurement error in label-free mass spectrometry. Because, as I said, we only consider the stochastic error between two replicates. However, for the systematic measurement error, we have no good way to measure that. Therefore, we look at the problem from another perspective. So there are two papers, one from Jonathan Weissman's group and one from David Bartol's group. They directly measure the translation rates by ribosome profiling. So here we are comparing their 3 direct measurements with the one in Schwanhauser, which is the black line. So we can see that all the 3 other direct measurements of translation have a similar distribution with a similar mean value in translation rates. However, for Schwanhauser, the distribution is more broad, and therefore the variance in the 3 direct measurements compared to Schwanhauser is about 4 to 7-fold less. Therefore, we think Schwanhauser overestimates the variance in translation. And by plugging, sorry, this is messy again. By plugging the direct measure rates of translation into our model, and without using the corrected protein abundance estimates. So here we are taking a different perspective. We use the direct translation rates, and we use the variance in measured protein levels in Schwanhauser. We didn't do the correction. And we come up with this result in the middle. So in this result, it shows that transcription contributes an even larger proportion than our previous method. So both our approaches provide some evidence showing that there may be some problem in the conclusion made by Schwanhauser et al. And so if you are interested in our paper, so we have publicly released it as an archive preprint, and you can Google my name and also the title, you can find the paper. And at last, I want to thank my former PhD advisor, Peter Bickel, in the Department of Statistics at UC Berkeley, and my collaborator, who has the main idea for this project, Dr. Mark Began, who is at Lawrence Berkeley National Laboratory, and I have collaborated with him on several projects. And for me, I have just moved to UCLA as a new assistant professor in statistics. Thank you very much, and I look forward to your questions. Yeah, I'm sorry that the diagram didn't show correctly. I don't know why. It's correct on my computer.
Questions? So what would you say you've learned from this that other researchers could then do to have better results in future analyses?
And can you repeat your question? Sorry.
I guess the question is, you determined a few factors that were problematic in this other study. So in terms of improvements to the methodology that would help other researchers in the future, what would you say the takeaway from this would be?
Yeah, I think our point is that we think it's very important to consider the measurement error, both stochastic and systematic, if possible, into the modeling without just using them as deterministic values. So this is our main point. And we think considering those errors may change the conclusion dramatically. Yeah, thank you.
So I would like to know your correction method for the Schwanhauser data set.
Okay, so for the Schwanhauser data set, so we have 66 housekeeping genes, which we have individual earlier measurements and the large-scale measurements. So we fit a spline model through it and use that to correct that. So we didn't show it here, but it's a very simple spline model. And we use cross-validation to verify that our model is not overfitting. Okay,
so then in the future, what should experimentalists do in terms of, for example, he wants to do in a similar scenario, what experiments should he do to correct those biases?
So for example, we said that in the Schwanhauser paper, they used 20 spiking protein standards in their label-free mass spectrometry experiments. However, those 20 spiking standards only cover the high concentration area. They ignore the low concentration area. So we think that in future experiments, it's very important that your spiking standards should cover the whole spectrum.
And for those mRNA biases, they should also do other kinds of experiments to control.
So for example, like here, the nanostring has been shown as a more accurate method, right? And comparing RSG to nanostring, we think is a way to help you at least have some estimate on the magnitude of the RNA error, measurement error.
Thank you.
Thank you very much. Thank you.
Okay, great.